Thanks, Dave. So Dave's already introduced me, but I'm Tristan. I am currently at the University of Reading in the Department of Meteorology. Although I'm not really a meteorologist by training, my background is in land surface remote sensing primarily, and I work on the land surface research component in the meteorology department. Currently, I am funded mostly by the European Space Agency, actually, to develop data assimilation systems for them. This talk is always a little difficult because from experience, we know that actually some of you in this audience won't, be planning to use or be particularly interested in remote sensing data. And other people will really be interested in using remote sensing data. So I try and pitch it so that it's going to be useful to everybody, and some bits are going to be hard for some people, and some bits are going to be easy for some people, but hopefully informative for everybody. Can I get a quick show of hands as to who is either using remote sensing currently or planning to use remote sensing data? Okay, so that's quite a few. Okay, so that's good. But what I want you to do is stop me, okay, and ask if there's something you don't understand. So I'm going to give a lecture now, which normally lasts anywhere between 90 minutes and 2 hours. We'll make sure we take a break in the middle. And then we're going to spend this afternoon actually looking at an easy method for downloading satellite data and using it in MATLAB and integrating it with flux tower measurements. Okay, so let's start. I stole this slide off Pat Morgan last year. So hopefully you used the same slide this year. But so last Monday, you were looking at that kind of scale, the stomata kind of level of carbon exchange. And today, we're bringing it right to the other end. And we're going to look at instruments that are capable of telling us things about vegetation on a global scale. We're less able to say explicit things about processes using this kind of data, okay, but it's useful because it allows us to examine how things are operating on a global scale. Okay, and if we want to know about how things are responding to climate, this is one really good test of that thing. So last week, you're using instruments like this. So you're all experts. In fact, you all know more about this than I do. At the moment, yeah, you can probably take one of these things and make some measurements of ACI curves. And honestly, I'd have to sit down with a manual for a day before I got that far. And today, we're going to talk about these kind of things. But the point that I want to make, the point I want to start with, is that essentially both instruments work in the same way. One's you've got in your hand and one's in orbit around the Earth. But the way they measure stuff is essentially the same. Electromagnetic radiation with a known distribution of energy is produced by some source. So in the light core, it's internal to the machine. And for remote sensing, well, it comes from a variety of sources, but what I'm going to talk about mostly today is using the sun as a source of that energy. That radiation interacts with a target of interest. And this changes the distribution of that energy in some way. So, in the light core, it's infrared radiation being absorbed by carbon dioxide. And we'll look at examples of how that happens for remote sensing. Some sensor records the intensity of the modified electromagnetic radiation in certain relevant domains. So, for example, in certain wavelengths or at certain angles or at certain polarities. And by knowing the distribution of the source radiation and the distribution of the modified radiation, we can infer something about what we're measuring. In the LICOR, it's a much, much more direct measurement of the thing you want to know about, because the whole system has been designed to kind of cut down on all the external uncertainties that affect these things. When we're working with satellites, we've got a whole bunch of other variables that we need to contend with to get to that information. So the problem is a lot more complex in a way. Classically, there are three types of remote sensing instruments. There's the passive instruments, this one and this one that takes reflected radiation from some source, which is generally the sun. Passive emitted remote sensing, which is generally thermal or passive microwave measurements, and active remote sensing, where the sensor has some mechanism for generating its own source. So radar and LiDAR are the main types there. I am almost exclusively going to focus on that pathway, the passive optical remote sensing using solar radiation. Now, partly the reason for that is because that's my expertise, but also this is the stuff that's kind of readily available to you and the stuff that's most obviously connected with vegetation and the health and status of vegetation. Okay? And it's kind of no coincidence that, it's because that's my expertise that led me to start working on ecosystem models and vegetation and combining these things together. This slide kind of stands on its own. I'm not going to come back to the information on this slide at any point. But it's here just to say that there is this whole problem in remote sensing of the sort that I'm talking about now. So where we have a path coming from, where our source of electromagnetic radiation is the sun, and we've got some passive sensor picking up reflected radiation, is that we've got some medium in between our target of interest and both the source and the sensor. And so the atmosphere is this huge problem, and it induces a whole bunch of noise and biases in what we observe. And I'm going to kind of conveniently forget about that for the rest of today. But if anybody wants to talk about it, I'm very willing to do so. I guess the thing to say about this is we have methods for dealing with that are pretty good nowadays. Not perfect by any stretch of the imagination, but pretty good. Oh, and by convention, that reminds me, by convention, over the next few slides, I'm going to be calling the intensity of the incoming radiation I0 and the intensity of the outgoing radiation I. So, the key to understanding the remote sensing signal is to understand how the target modifies that distribution of the incoming electromagnetic radiation. And as ecologists, what we are primarily interested in, or as ecologists, sorts of ecologists that would take this course, what we're primarily interested in is how that radiation interacts with plant canopies. Now, what we're going to do now is a little bit complex. Hopefully, this follows on really kind of nicely from Dennis's talk. So I've seen Dennis's talk, and this kind of extends on the things he was doing. Now, what I haven't seen is exactly what Dennis talked about this time. Okay, so if this isn't making any sense, stop me. So let's assume that we have some plant canopy and it's filled with randomly positioned plate absorbers. And by that, I just mean leaves. And I'm ignoring trunks of the plants or anything like that. So we've just got leaves. So we have a plant canopy like that. And that is, believe it or not, pretty close to the approximation that most models make about plant canopies. So most ecosystem models assume that that's what the canopy is like. Dave described this as a green smear just 20 minutes ago or something. Actually, I nearly corrected Dave as he was talking. It's not even a green smear, it's a black smear. I'll talk about that in a moment. My first variable that I'm going to talk about is the depth through the canopy. Okay? So I'm introducing a variable that sometimes is called Z. I've also tried to make my notation as close to Dennis's as possible. There's a particular note where I've not achieved that. So let's consider a single photon traveling through that plant canopy at some angle over a certain path length. Okay? That's straightforward. And this angle here would normally be the angle corresponding to the angle that the sun is at, unless that photon happens to have gone through some other scattering event before arriving at the canopy. But we're going to ignore that for the moment. So basically a photon coming in at the angle that it left the sun at and passing through that canopy. Okay, so we've got the angle and delta M. Actually, this is a two-dimensional angle, but because I'm on a flat screen, it's essentially that zenith angle. Right. The most important thing that we need to know, first off, for thinking about photosynthesis and all those kind of things, is what the probability of that photon being intercepted in the plant canopy is. So we have some probability, and it is dependent primarily on that angle and the amount of distance that we're considering through that plant canopy. So it's probability, P, as a function of angle and path length. And we have a very, very simple expression for that. The probability is equal to the leaf area density within the volume that it's passing through, and that is not a leaf area index, okay, which is probably what most of you are more familiar with. If we integrate across the volume, we get the leaf area index. So the probability is equal to the leaf area density at some position, which we're just going to call R arbitrarily for the moment, and some function of the way in which the leaves are projected, which generally in the kind of plant radius of transfer community we actually just call the G function. Now, to explain that, think about how likely it would be that a photon would be intercepted if all the leaves were facing the photon flat on, or if all the leaves were facing the photon point on. Okay, you're much more likely to be intercepted if all the leaves are like that than if they're like that. If the leaves are all randomly distributed, the G function is just 0.5 in all angles, basically. Important note. Is that this then is used to represent the clumping factor in the canopy. So Dennis used omega to represent clumping, and I'm using it here to represent the angle of the photon traveling. Okay, so if the intensity of the incident radiation field at some depth through the canopy Z is given by I, then the intensity at the depth Z plus delta Z is given by the following equation, right? Now, these things are just kind of by definition. So I'm not adding anything new here. I'm just saying we've got some intensity at this point traveling at a certain angle. And we are going to consider the intensity just at a slightly smaller depth in the canopy, intensity of radiation. So we know the intensity at some point, and we want to know what it is just below that point in the canopy. So all we need to do then is multiply the intensity, the first intensity, by the probability of it being not intercepted. Okay? And we have this term on the previous slide. Now, hopefully that all makes sense, that basically you're using the probability of interception to calculate the intensity of radiation at a point just below the first point you considered. And we can sub in those terms that we had previously from our definition of P. So the leaf area density, the leaf angle distribution function, and our path length. So schematically, we have this set up now. We're considering the change in intensity from this point in the changing the intensity of the radiation from this point in the canopy to this point in the canopy over some path length given by delta M. Is everybody reasonably comfortable with that so far? That's good. Okay, so everything I've described so far is very, very general, okay? And we can use it to build quite complex models of light interception in plant canopies. What we're going to do now is start removing some of that generality from the model to try and actually build something that we can turn into a real, an actual equation that we can describe something in the canopy with. So the first assumption we're going to make, which is very, very common in all sorts of ecosystem models, is to assume that the canopy is plane parallel, okay, and homogeneous within that medium. Now, all that means is that it has a top and a bottom that are basically parallel with each other, and inside it there's just randomly distributed stuff, leaves. In that case, we can describe the path length by the depth divided by the cosine of the incoming angle. So all I've done on this slide is introduce an expression for the path length as a function of the depth, the change in depth that we're considering, and the angle that the photon is traveling at. And we're allowed to make that assumption because we're assuming that the canopy is plane parallel. So we now drop delta M from our equation and replace it with delta Z mu, where mu is the cosine of the incidence angle of the radiation. Okay? Okay, so what we'd like to know, rather than what the intensity is at that new depth, is how the intensity changes, okay, with height in the canopy. So that's simple. We just subtract the initial intensity from both sides, okay? So the change in intensity, the first point we were considering becomes 0, and we get the actual, rather than the intensity at the delta Z, we get the change in intensity at delta Z, okay? So simply subtract from both sides, and we end up with that equation. Now, hopefully that's clear that we've got rid of that 1 minus, yeah? But, whoops, wrong way. Now, we can now consider that in the limit, Okay, so calculus, which everybody loves, and setting the limit of delta z to 0, so as the change in the height through the canopy tends towards 0, we can formulate a differential equation for that change. So change in intensity with change in height is equal to that. And we have lost here that delta z mu term. Okay? Just basically come over here. If you're, so at some point, some point during me doing this derivation, some people will drift off at different points, right? And some people will stick with it through to the end. But what I hope I'm doing here is setting you up with a kind of clear enough description that you can go back to it and think back through it at some point. Now, we're in a position to integrate that equation. So we can take any 2 points in the canopy and integrate across those positions. And the integral of an equation like that is going to have an exponential in it, and it just turns out that we get this expression. And because we've integrated across our volume, and we had originally this leaf density function as a function of its position in the canopy, we lose that, so I've integrated r out basically of the equation, and that becomes a leaf area index. So we have the leaf area index, okay, over some depth of the canopy z, times by that leaf angle distribution function, divided by the cosine of the incident angle of that photon coming in. So if you then multiply that by the intensity of radiation at the top of the canopy, so we've got z equals 0 here, we get the intensity at any depth in the canopy that we're after. Typically, what we want to know, rather than the transmission, is the absorption, and that's a simple rearrangement of that equation, and we end up with this, where I'm now calling F is the fraction of absorbed radiation, and I've basically divided through by the intensity at the top of the canopy, so we get a 1 here. And probably most of you are familiar with that equation. Maybe not quite in that form, but that's Beer's law. And it's what a large number of ecosystem models use to describe the interception of light in the canopy. Everybody kind of comfortable with that? And that's kind of what Dennis was probably talking to you about on Friday. Those kind of representations. Okay. Now, this is what we've just described is about interception in the canopy, and not, we've assumed, we've made this implicit assumption in that derivation that anything that's intercepted is absorbed. We sometimes refer to that as the black leaf scenario. Black just meaning that any light hitting it is absorbed. So it assumes, there's an implicit assumption in what we've just done, that there's no reflection from the leaves and there's no reflection from the background, so the soil typically. So it's the black soil, black leaf case, and that is what most ecosystem models have in them. Almost all of them contain those assumptions. There's certainly all the simple ones. Actually, I'm being a little bit unfair. It's getting better, okay? I'm being a little bit unfair. But so SIPNet, which you'll use later this week, contains that assumption. The Sheffield model, which is a big global scale model that I use a lot, also contains that assumption. And in fact, it's not uncommon to have models that are capable of predicting the albedo of the surface, the plant canopies, but still have that assumption embedded inside their FA par routines. Okay, so they still predict something about the reflectivity of the plant canopies, but that energy they're reflecting does not balance with the energy they're absorbing. So the big problem for me, as somebody who tries to work getting satellite data into ecosystem models and models of carbon fluxes, is that that representation tells us absolutely nothing about what a satellite sees, because if we use what I've just described to predict the satellite signal, basically we have everything as being black. And that helps us in no way interpret the satellite signal. In other words, the physical representation, In other words, the physical representation of light scattering inside many canopy models is not sophisticated enough to represent observations from remote sensing. And actually, even those models which are starting to include leaf reflectances and things like that are still not really sophisticated enough to predict remote sensing signals. There's a whole bunch of things we have to do to make that happen. And the most obvious ones are the ones that I've been talking about. So we would probably want to have some element of radiation that's scattered by the soil, going back up to the satellite. Obviously, some stuff hitting green leaves and being bounced back up to the satellite. But what's often not so obvious is that we need to have vegetation that kind of clusters together in discrete units, because in the real world, vegetation doesn't exist in some plain parallel homogeneous media. It's often the case that when we use a kind of Beer's law representation to estimate the fraction of absorbed radiation in the canopy, that what we're doing is we're calibrating that relationship to kind of implicitly contain that level of clumping. But actually, we need to be explicit about it if we want to represent what the satellites see. Another way of looking at this is to consider that there's some energy budget in play. And so we have the downwelling intensity of radiation, and that is equal to some amount of energy that's reflected plus some amount of energy that's absorbed in the canopy and some amount of energy that's absorbed in the soil. And those things have to balance. We know, or we assume we know pretty well, the intensity of the incoming stuff. We have that atmospheric problem going on, but let's just assume that we have a handle on that. We measure reflectance, the reflected portion of the light. So that's all that happens with the remote sensing instruments, is that we make a measurement of that. Although it is important to point out that we don't measure the whole of that R for this energy budget representation. We measure it at some given wavelength and at some given geometry, so at some angle that the satellite's in. We don't measure the integral across all wavelengths and their energy excitant in all angles. But nonetheless, that is a measurement. So we've got something we know and something we measurement. And actually, we are pretty confident, if we're talking about knowing what's leaving at the top of the atmosphere, remote sensing measurements are pretty accurate. Keith, it's fair to say we know the energy incident at the sensor, we know that pretty accurately, right?
Yeah, and once again, if it's a satellite, we know it better than if it's, say, like an airplane, maybe the atmosphere above you, they'll take it
Yeah.
down.
Now, what we want to know, then, is what's absorbed, generally speaking to this audience. It's not always the case that what everybody wants to know is what's absorbed in the canopy, but I'm speaking to this audience. What we want to know is what's absorbed in the canopy, or perhaps things we can infer from what's absorbed in the canopy, you know, like chlorophyll content, leaf area index, so on and so on. But it's not necessarily at the same wavelength or geometric setups as the thing we observe, which complicates matters. And also we have some amount of radiation that's absorbed in the soil, which is difficult to account for. And it's complicated additionally because we can kind of rewrite the energy budget for the reflected component as having a bunch of different stuff in it. One common way of breaking that down, and we're going to stop getting, I'm going to stop being abstract in a moment so you can kind of breathe a sigh of relief. One common way of breaking that R term down is to consider the component of that that has a single interaction with the canopy, the component of that that has just a single interaction with the soil, the component of that which has multiple interactions with the canopy alone, and the component of that that has multiple interactions with all of the media, so the canopy and the soil. Now, that means that just knowing R, even though we know R well, you know, that is based on a whole bunch of other things that includes interaction with the soil. Okay? So if we want to infer information about that thing we measure, we need to understand all of those things. So think back to that FA par equation and all the stuff that Dennis talked about on Friday, and all the stuff I've just derived. And this is what I would say is probably the simplest set of equations you can use to describe the remote sensing problem, rather than the Beer's law problem. And it's the reason why I select this is A, because it's relatively simple, and B, because it uses that breakdown of the energy budget that I just described. I'm not going to go into details, we're not going to derive any of this, we're not going to talk about any further, but that paper is there for, you know, if anybody wants to follow this up, I suggest that paper is an excellent place to start. That first term is the single scattered soil reflectance, so photons that just go straight through the canopy, don't interact with any leaves, bounce off the soil, straight back through the canopy. So that one's obviously more important when you've got a thinner canopy, yeah? So fewer leaves, that term becomes dominant. The next term is the single scattered canopy reflectance, so things which just hit a leaf and bounce straight back out. So this becomes important as you get a stronger canopy. And these two equations are fairly explicit. That is more or less all the maths you need to explain those terms. Okay, there aren't many other kind of functions underlying those things, just a couple, but. And then this last term represents the multiply scattered reflectance. So things that interact multiply inside, photons that interact multiply inside the canopy and between the canopy and the ground. This actually is not so explicit. There's a whole bunch of other stuff underlying this, and it requires numerical solution to solve this. Okay? So this is where it gets a lot more complex. Although actually, if you've got a canopy with relatively dark leaves and if you've got a relatively dark soil, this, you know, you can sometimes get away with ignoring this. A first order approximation would be given by just taking the first two terms. So what we now have then is kind of a new way of looking at the problem, the light interception problem, that gives us some handle on what the satellite's seeing. Okay? And at first glance, that would appear to be really poorly constrained, because what we've added in is a whole bunch of other stuff into there, and we've just got this observation of reflected energy. But the good news is that remote sensing data has a lot of diverse types of information hidden in there that allow us to kind of solve those equations. So we have information at different wavelengths, which is what I'll talk about mostly over the next hour or so. Information at different polarizations, which I'm not going to talk about so much at all, in fact. Angular information, BRDF, which is where I kind of cut my teeth. That was what my PhD was on, was how you can use observations at different angles to infer information about the canopy. And then kind of more obviously, we have spatial and temporal information. That might sound almost trivial, but actually there are some important things you can do with that. The remote sensing problem then is to exploit those sorts of information to provide those sources of data to provide information about the target. Right, so all of you know this, I think, probably. This is kind of, you know, this is high school stuff, basically. Electromagnetic radiation. Different wavelengths going in this direction. And this is kind of a rough schematic to show you what the frequency is and what the colours are that we see. We also have, interestingly, only selected regions where this stuff penetrates the atmosphere. So it restricts us to what we can, the wavelengths that we can use to do these things. And I'm going to focus exclusively on this region here today and kind of ignore the microwave stuff, although I'll say something very, very briefly about that at the end of the talk. Just to put it into context, this is the stuff we can see. Okay, this is the visible range of the electromagnetic spectrum. And that, you know, comprises probably the most readily available sources of data that you can get your hands on to do this kind of work with. Now, the bottom, let me go back actually, the very bottom bar here is the temperature of an object that emits that radiation, the temperature of a black body, so an idealized emitter that emits the frequency here. So this stuff here is at about 6,000 Kelvin, which is the temperature of our sun. Okay? So the visible wavelengths of light are emitted by a body that's about 6,000 Kelvin, and that's approximately the temperature of our sun. I've actually forgotten which textbook I photocopied this from, but this is from some old classic geography textbook. And that curve over the top is the idealized blackbody curve. So this now is wavelength on the x-axis and basically the emitted energy on the y-axis. Underneath it, the solid line underneath that is the solar radiation that actually reaches the top of our atmosphere. So pretty close to what you would expect for a 6,000 K blackbody. And underneath that is the amount reaching the Earth after interactions with the atmosphere. Putting that into context, those are very approximately the wavelengths of light in which we see. The blue, green and red parts of the electromagnetic spectrum, and they're at that peak of the blackbody curve for 6,000 Kelvin. Now, it's probably no coincidence, it's not probably no coincidence, it is no coincidence that we see in that region of the electromagnetic spectrum. We've evolved to utilize the part of the energy spectrum that's most abundant on our planet, okay? And plants have done exactly the same thing, of course. They absorb in the red and blue parts of the spectrum because that's where most of the energy is. It would be a crazy strategy for a plant to develop some, you know, alien photosynthesis mechanism that used this part of the spectrum because it would have no energy to do its work with. And it's kind of because of that that optical regions of the remote sensing instruments are useful for our plant studies, because those are the parts of the electromagnetic spectrum that, you know, most things have evolved to use. So really a lot of what I'm going to talk about today is just restricted to that limited range of data, that wavelength range. The nice thing, from some perspectives, is that it gives us an intuitive kind of set of data to work with. And this is an example of data from the MODIS instrument, which flies on board 2 NASA satellites, the Terra satellite and the Aqua satellite. And I've picked this image for no other reason than it's nice. But it gives us a feel for the kinds of information that you can see in that data. There's this lovely phytoplankton bloom down here, lots of photosynthesis going on, and a whole bunch of detail on this small island, and I'm afraid I forget exactly where it is. There's a bunch of detail on here that, you know, gives us this tantalizing image, of a tantalizing view of the sorts of things we might be able to infer. You can really clearly see where there's snow. You can really clearly see how dense the vegetation is at different points. You can see the hydrological routing going on. So there has to be things in there that are going to tell us about ecological systems. Now, I am going to devote a lot of time now to talking specifically about that sensor, about MODIS. How am I doing for time, by the way? Has anybody got, I don't have time on me. 10:35, right. We can keep going. So I'm going to devote a lot of time to talking about MODIS. So we've I've done my first bit, where I wanted to kind of introduce some of the kind of theoretical stuff, give you some feel for the problem that we're faced with for connecting the assumptions in ecological models and the observations we make here. This now then is more general. I'm going to talk about MODIS because it's readily available. You can all get your hands on it. I'm going to teach you how to get your hands on it this afternoon in a nice, simple way. And importantly, there's been a lot of energy devoted to delivering products from these sensors that are relevant to carbon cycle science. So, for example, there are photosynthesis products you can get from MODIS that you can download straight away and use them. Okay, so in deciding on something to talk about for this lecture, it seemed to me that the best place to go was talk about these kind of things because it's something that all of you have the potential to use. And even if you don't use it, these kind of things are being more and more widely used in the literature, and having some kind of understanding about how they're produced and what their limitations are, I think is valuable to all of us in this community. Okay, so MODIS, there's two of them in orbit, one on board Terra, one on board Aqua. They've been in orbit since 2000. Terra MODIS was launched in 2000. Aqua was 2002, I think. Correct me if you know better. You get daily global coverage from the instrument, which is a hugely valuable thing, and with its finest spatial resolution of about 250 meters. Now, for some applications, that's way too coarse, right? But actually, if you're talking about the footprint of a flux tower, that's not so bad, 250 meters. It has a whole bunch of spectral channels, some of which are specifically devoted to land surface studies. I'll describe what those are in the next couple of slides. The other thing that the nice people who work on MODIS data have done for us is produced very thorough, or comprehensive perhaps is a better word, set of quality control information that comes with the data. It describes the likely sources of error in that data. So, for example, where they haven't had enough information, enough observations to generate a specific product to a certain level of quality. And importantly, it allows users to filter those data out. There's extensive validation activities underlying this sensor. The last decade has been almost like a golden decade for remote sensing of this sort. Lots of peer-reviewed literature describing errors in the data and online information. I'm not going to talk about that anymore today in this lecture because, it's really not that interesting. So instead, we're going to talk about it a lot this afternoon. But it is. So for those of you who are never going to use this data, that's kind of not so important. For those of you who are going to use MODIS data, understanding the quality control information is vital. So, our basic source of information, the kind of the first order source of information from remote sensing instruments, if you ask most people, they would say it was spectral information. And so by that I mean the reflected portion of electromagnetic radiation as a function of wavelength. And the nice thing is that different surface types have characteristically quite different spectral signatures. So these spectra are all modelled, but they're modelled with a reasonably high degree of physical accuracy, shall we say. And actually this green line and all the canopy spectra that I'll show you I've modelled using the semi-discrete model, the maths of which I showed you earlier. But I think what's very clear from this slide is that if you had, you know, a world that consisted of snow, vegetation and soil, we'd be able to distinguish between those three things pretty easily just by perhaps 2 spectral channels, okay? We can see those differences quite clearly because our eyes are sensitive to these parts of the electromagnetic spectrum. So it's very approximately blue, green and red parts. And so actually the vegetation line doesn't come out very well there. But, you know, you can see clearly that we've got that green peak in the vegetation, which distinguishes it quite nicely from soil, and obviously snow, very bright in all the visible wavelengths. MODIS, the optical channels in MODIS are roughly positioned here. Notice that the numbering scheme of the channels does not correspond to their progressive, it doesn't increase with wavelength. It's actually slightly to do with the resolution. So this is red and near infrared, and they're the two that generally get the finest resolution. That's right, isn't it, Keith? They're the 250 meter channels. So we have 1 and 2, then 3, 4, 5, 6, 7. And we have a whole bunch of other information that's available to us because we have those different spectral channels. So as well as seeing approximately in the red, green and blue, just like our eyes do, we get the near infrared out here, and you can see there's a big distinguishing feature between vegetation and soil there. Out into the shortwave infrared, where things start to get sensitive to water content and so on. To illustrate that more clearly from the point of view of vegetation spectra, what I've done is modelled a bunch of spectra with a plant canopy with different conditions. So a plant canopy with a high leaf area index, plant canopy with a very low leaf area index, plant canopy with very low chlorophyll content in the leaves, and plant canopy with low water content in the leaves. And other than that, I've kept all the other variables of the system the same. So if we take the high LAI canopy and the low water canopy for the moment, so that's that big thick red line is the healthy high LAI canopy, and the low water canopy, and by low water I don't mean so low that it's started to senesce or anything, it's still got nice green leaves. You can see that in the visible parts of the spectrum they're identical, those two canopies basically. But once we get out into the shortwave infrared, we have a mechanism for distinguishing between canopies that are water stressed and canopies that have got plenty of water. And it almost doesn't make any difference whether our plant has low LAI, low chlorophyll or high LAI. What's making the difference here is whether or not it's got a low water content in its leaves. Of course, I'm simplifying things massively here because obviously the soil's going to have a big impact. The soil moisture content will have a big impact in this part of the spectrum and so on and so on. But I think you can start to see, hopefully, that by having those kind of that range of spectral signatures, we have information where we can start to, data where we can start to tease apart some useful information about the plant canopy. I'm wondering how much skepticism there is in the room. Right. So, nice thing from our point of view about using the MODIS data is that people have invested an awful lot of money and time in sorting these things out for us already. Okay? So it's one of the reasons why it's worth talking about MODIS and worth learning to use the data, potentially. So this is an example of the MODIS leaf area index and fraction of absorbed photosynthetic reactive radiation product over South America, the northern half of South America. It's a physically based algorithm, by which I mean it has all the scattering laws that I described earlier that you would need to represent the remote sensing signal. And what it does is uses 8 days of surface reflectance data from MODIS to invert that model, to take the observations and work out what the parameters are inside the model that give the best fit to those observations. And the main parameter that it extracts is the leaf area index from that model. So by doing that inversion procedure, we end up with an estimate. We start with the reflectance data and we end up with an estimate of the leaf area index. It also estimates some of the structural components of the canopy, which then allows us to get, along with the leaf area index, allows us to get the fraction of absorbed photosynthetic reactive radiation. But because this algorithm considers those structural components and has leaves that reflect light in a way, the LAI you get from this algorithm will not give you the same FA par as it would if you just plugged that LAI into Beer's law. Does that make sense? The rough shape of that relationship would be about the same. You get the same kind of exponential growth in FA par with LAI. Okay, but exponential decay. But it's not simply a restatement of Beer's law. It's not that 1 minus e to the minus k LAI. This is from the algorithm theoretical basis document for the leaf area index product, and it gives us some idea of what happens to the data to generate those products. And I'm going to spend more time discussing this for the photosynthesis product, but it's worth kind of having a look at this one because this data actually feeds into the photosynthesis product. So really the main thing I want you to take from this map is there's a whole bunch of complex stuff that happens to that reflectance data to turn it into leaf area index. And really, all this other stuff that happens isn't data as such, right? The stuff that we observe is this. And then it goes through all this other transformation to get to leaf area index. We have some uncertainty because we have that problem with the atmosphere and we don't know surface reflectance perfectly. So we've got some uncertainty going into the product from there. It also relies on a biome classification map, which is also actually derived from MODIS data. And that also has a actually fairly high level of uncertainty associated with it, certainly comparative to the surface reflectance product. So 2 sources of data uncertainty going into that product. There's also the RT algorithm, which is the radiative transfer algorithm, is the way that the physics of the photon scattering in the plant canopy is represented inside the model. And of course, no model, like Dave said earlier, is perfect. And it has to make some simplifying assumptions just to make it a numerically tractable problem. So there's some model uncertainty inside the production of these data as well. But there's been quite a lot of effort devoted into understanding what those uncertainties are like. And this is, I think, the most up-to-date information we have. Official line. There are lots and lots of papers on the validation of these products out there. You go onto Google Scholar, type in, modus LAI accuracy or something like that, you'll get a whole bunch of papers. But so the official line, and I'm fairly sure this is the most up-to-date one, is that the accuracy is about 0.66 LAI units, RMSE, when all biomes are taken into account. So that's kind of like the average error across all biomes. It will be, generally speaking, more accurate for less dense canopies. So it's probably going to do better for savannas than it will do for rainforests. And the accuracy in FAPAR is about 0.12 root mean squared error. Now, that's like saying you've got a 12% error on your, well, not an absolute or relative error. So, if you had a canopy of which was absorbing about half of its incoming radiation, approximately, then you've got like a 20% error on the amount you're absorbing, and that, in terms of photosynthesis, is going to make a big difference. I'm going to show you some examples of the data in just a couple of slides, and I actually, I think those figures might be just a little bit optimistic. The caveat, the thing that you need to keep your heads around, is that there is an exponential function between leaf area index and FAPAR, and if you go back to my slides from the beginning, you can see that relationship come out of the derivation that I went through. And so this now is, all of these curves are for some forest canopy that's plane parallel and has all those assumptions in it, but I'm modeling all the stuff that I need to know to calculate satellite reflectances. And they're all the same, they're all identical, except that I've varied the LAI from 0.1 to 15. Okay, so a very, very fine canopy to an almost impossibly dense canopy. And the thing that I want you to notice is that between an LAI of about 7 and 15, there's virtually no difference in the reflected radiation. So it's certainly at that kind of level that's within the scope of our ability to observe those reflectances. So there are going to be big errors when you have high leaf area indices. But for FAPAR, because the relationship's exponential, the errors are kind of more evenly spread about those things, more linear across that range. Now, this is what the reality looks like. This is for Niwot Ridge, and it's Niwot Ridge Flux Tower and a kind of 3x3 pixel sample around the Flux Tower. And to be fair, I have not applied any quality control to this data. So I'm kind of exaggerating it a bit. So, and we will spend a lot of time looking at the quality control information later. And also, it's also fair for me to say that they've done a lot of work improving the LAI product since I first made this slide. But look at the variability in the predicted leaf area index. We're going from LAIs of 6.5 to 0.5 in the space of a few days. Now, probably, if I applied the QC, it would take this low peak down, but it would still be moving up and down quite a lot. And it's not unusual to see movement in the leaf area index of 2, an LAI of 2, up and down between samples. The leaf area index and the FAPAR, they come bundled together as one thing. And the corresponding FA-PAR data looks like that. So slightly better when we've got a full canopy, okay, but worse when we've got a low canopy because it's more sensitive to those errors. Has anybody noticed something that's really wrong about both of these plots? So they're both very noisy and spatial consistent, you know, in both temporally and spatially. Spatial consistency is poor, temporal consistency is poor. It's an Iwat Ridge. What is really wrong about both of these plots? It's an evergreen forest. Right. And yet there's a clear seasonal pattern of leaf area index. Dave and I were doing some work about 4 years ago or so, and we noticed that there's a really strong correlation between the seasonality in the forests around here, as observed by the MODIS LAI and FA-PAR product, and the timing of snow melt from there's a snow network, snow observing network called SNOWTEL, I think, in the Rocky Mountains, and basically we were able to correlate snow melt in SNOWTEL basically with the apparent green up in this evergreen forest. So it's really sensitive to things. Oh, and in that study we had applied all the quality control flags correctly and as rigorously as we possibly could, and it still had this seasonality in the forest that you just wouldn't expect. What another thing that comes out of the MODIS data stream, and it follows on from the whole leaf area index FA-PAR thing, is estimates of photosynthesis, and actually also annual MPP they do too. Okay, well, we've already seen everything that goes into that leaf area index and FA-PAR products, and then they take that data and they plug it into the photosynthesis products. And Debbie Huntsinger might talk about the accuracy of this a little bit because she's done a lot of work on intercomparisons of projects.
What's the resolution of
1 kilometre in this case. So it's kind of at the scale that you might be able to compare it reasonably with a flux tower. And it's quite commonly done to compare this data with a flux tower stuff. And in fact, we might do that, well, we will do that this afternoon. So if you're looking at kind of broad spatial patterns and temporal patterns of photosynthesis, it looks pretty believable, actually. In some ways, this kind of smooths out some of the problems with the leaf area index product. But of course, really what we're interested in is whether or not it gets the magnitudes correct. The photosynthesis model, and Dave alluded to this in his talk, is basically, I'll show you the full schematic in a moment, but it's basically this. Photosynthesis is equal to FA-PAR, the fraction of absorbed PAR, times by PAR. So basically those two terms there, FA-PAR times by PAR becomes absorbed PAR, and then just multiplied by some notion of the efficiency of that ecosystem or that biome of turning that energy into carbon. You know, so the units of that efficiency term are literally grams of carbon per megajoule of solar radiation. It's typical for, however, for that efficiency term to be some sort of model. Well, in the MODIS product it is. In some of the other products, not. But for MODIS, that efficiency term is a simple model, which is basically that each biome will have some hypothetical maximum efficiency, and then that is scaled by a bunch of functions of things like temperature and vapor pressure deficit. And what's more, what's important to keep in mind is that those functions, those scaling functions, and they do just scale that hypothetical maximum. They go between 0 and 1. Can you see that? are very, very simple functions. Very simple. There's no real kind of understanding of the physiological processes underlying that description. So basically, on this slide, so this is the temperature function, this one here, and it's basically saying if you have a temperature that's below some threshold, then this efficiency is going to be 0 and you'll get no photosynthesis. And if it's above this threshold, you'll get this will be 1 and you'll have your full efficiency. And between those points, it's just a linear ramp. Okay? So simple, simple. This is the schematic of the whole product. And actually, let's ignore, really, you can ignore this middle box. This is what they do to estimate annual MPP from the data. And what I'm going to talk about really is the GPP, the photosynthesis part. And the satellite data, the stuff that we're actually observing, and remember right back at the beginning, I said that that LICOR gives you a much more direct observation, okay, of CO2 exchange at the leaf level than this. The satellite data in the photosynthesis products is here, okay, and only there. And you've got to do all these other things to it to get photosynthesis. And then cast your mind back to a couple of slides ago where I showed you the schematic of the FA-PAR product, and there was all that other stuff happening to that to translate the satellite reflectance data into FA-PAR. And then you've got to push it through all of this stuff again to get it to photosynthesis. So actually, the photosynthesis estimates are really divorced from the actual observations that are being made by the satellite. That's a really important point that I want to get across. The red boxes then are where the satellite data is coming in. The green boxes are where there's some lookup table being employed. And so that might be, for example, a lookup table as a function of the biome that is being observed. So the hypothetical maximum efficiency is based on, whether it's an evergreen forest or a deciduous forest or so on. And the blue stuff is where there's some kind of external climate data being employed. So minimum temperature, vapor pressure deficit, and so on. That all comes from external sources to drive this product. And the accuracy of the products is, to a large degree dependent on that. Now, there's actually, in some ways, there's nothing wrong with doing that, but what you need to keep in your minds is that there's an awful lot inside these data that is not the satellite data. And actually, I think a particularly important point that I can't stress enough is there's modelling that goes on in there. There are some models underlying those things. So those very simple models of the response of photosynthesis to temperature. It's a model that exists in there, and that model has assumptions in it. So, some caveats to the using this data then. It inherits errors already present in the leaf area index and FA-PAR product. So before you get any errors in this, it's already inherited a bunch of noise from the stuff that comes before it. It has driving climate data in it. It uses very, very simple models for efficiency, so linear ramps for vapor pressure deficit and temperature. And it's driven by generalized lookup tables based on only 6 vegetation types for the whole world. It's almost amazing how well it does when you consider all these things. I mean, it doesn't do brilliantly, but we'll actually have a look at that this afternoon to see how well it does. And when you consider all this, you feel a lot better about the data when you see it. When it doesn't match your flux tower so precisely. This then is the corresponding plot to the leaf area index and FA-PAR stuff I showed earlier for the MODIS GPP product. And you can see the same kind of things. it's noisy, not so consistent. But actually, we can actually now believe that there might be some seasonal pattern to GPP. And of course, one of the things that happens here is that because it's driven by PAR data from some external source, it imposes that seasonality onto the products already. Does that make sense? You've got the seasonality of the incoming radiation, which has nothing to do with the satellite data, which is actually controlling a lot of the seasonality you see in the GPP product. Okay, so my important point number one was about the assumptions we make and the difference in the assumptions you have to make if you want to do work with satellite data compared to those assumptions that typically underlie ecosystem models. The point that I want to stop and reiterate now is that Modis has all these readily available data products, and they are kind of useful things to have. It's a valuable service that they've done. And you know, if you look at the volume of papers that have been produced using these products, it is enormous. But it's really important to be aware of, in my opinion, two key caveats. There is some inherent noise in these products because they try to invert physical models using relatively very small amounts of data that have noise in them. It's not a particularly well-posed numerical problem, so there's an inherent level of noise in a lot of the data products. However, in some ways that's practically the most important thing, just to be aware that there is noise in these data. I think the most important thing, however, on a kind of philosophical level, if you like, is that the assumptions used in the derivation of these products are not necessarily consistent with the assumptions in any given model. Okay, so we saw the GPP product and it's driven by some climate data. If you then compare it with the outputs of some model that you've just run, that's probably also driven by some climate data. And probably those climate data are different. So just because the two for your model and the Modis photosynthesis product, taking that as an example, don't match up, okay, it doesn't necessarily mean that your model's wrong or that the data's wrong. It could just be that your climate data are different. Your climate data could be wrong. There are also different assumptions about, for example, the temperature response to, the temperature response of photosynthesis. Okay, that will be different inside the Modis product and inside any given model. So really, to use these products intelligently, you have to think about how those things are represented inside the product and the model you're using. I think that it is one of the key barriers to successfully using these data assimilation projects using ecosystem models, basically, is that we are very bad at characterizing the errors. And also, if you try and assimilate these data into models, you're not just assimilating the data, you're assimilating a whole bunch of assumptions that might conflict with the assumptions inside your model. Right. That's it for those kind of Modis product things. I want to give an honourable mention to vegetation indices, because for some of you actually they may turn out to be a really useful tool. And I was as I did my PhD research, I was taught to hate these things, you know, to loathe them with a passion, because they're not very, well, there's no real physical basis for them, okay? They're broad brush indicators of the amount and status of vegetation in some given area. But as the years have gone on, actually they're quite useful for some things, it turns out, as long as you're kind of prepared to accept their limitations. Now the basis of most vegetation indices, and I'll explain what they are in just a couple of slides, is that vegetation exhibits a high degree of difference, healthy vegetation exhibits a high level of difference between the red part of the electromagnetic spectrum and the near-infrared part of the electromagnetic spectrum. So if you look at the thick red line, which is the healthy high LAI canopy that I introduced earlier, the reflectance in the red part of the spectrum is very low, and the reflectance in the near-infrared part of the spectrum is very high. And if you then take a canopy that's not doing so well, so this is the dark blue line here is a low chlorophyll canopy, the difference between the red and the near-infrared part of the spectrum is much smaller. Also, if you think about the way soil works as well, that would have that's less different as well. So if you've got a lot of soil present, you can use them to, it will distinguish that situation basically. The calculation is trivial. It is simply near-infrared minus red reflectance over the sum of near-infrared plus red reflectance. That gives us, that equation for any set of numbers will vary between -1 and 1. But generally, for most of the land surface, it will be greater than 0. Because for most of the land surface, the near-infrared reflectance will always be higher than the red reflectance. It is broadly related, this relationship is broadly related to the amount of vegetation present. But it's very difficult to do anything really quantitative with it because it's affected by a whole bunch of things like soil brightness, the atmosphere, and so on and so on. And there's been a whole bunch of variants introduced to kind of counter these things, but really none of them are especially perfect. You can see this is a global map of NDVI from some point in time and a conveniently broken scale bar, but you can see that it's representing the kind of the spatial variability and what you might expect of vegetation across the world. So, there is some valuable information in there. So here's an example of how I've used that in the past, and this paper was looking really at what was limiting plant growth we basically wanted to establish relationships between climate variables and vegetation in Ireland. Actually, Dave's gone, unfortunately. I'm talking about his homeland. And what we did was to use NDVI and show that it has a really strong correlation in Ireland with temperature. So we could deduce from that the Irish landscape is basically a temperature-limited environment. We were looking at the limitations on photosynthesis for that part of the world. We're not using it to infer anything quantitative, just to say that, yeah, we think that because we've done this piece of analysis, that we can say that it's those kind of factors that are limiting plant growth. Does that make sense? Not trying to infer anything quantitative. It's a very, very small part of a much larger paper. But if you try to do that same analysis with the leaf area index products, there's a whole extra bunch of processing you would have to do and stuff. So this was like a very quick look at how you might do that. And just to kind of illustrate that for some purposes, it might be a better thing. That is the NDVI profile for the same point that I had those, those LAI and GPP and FA-PAR plots for. And it's a much, much cleaner signal. There's a lot less noise in it. Okay? So really, if you wanted to detect the seasonal cycle, you'd be much better off using that vegetation index than you would the leaf area index product. Modis actually very popularly ships this thing called the Enhanced Vegetation Index. It looks like that. It's basically the same thing. It's based on the difference between the near-infrared and the red reflectance, and it's designed to reduce atmospheric noise and corrects for variation in soil brightness, and it is claimed that it is more closely related to FA-PAR, but honestly, that's up for debate. This is work that comes out of Boston, actually, and it's a big analysis of the relationship between NDVI and leaf area index. suggesting that there is some, there is some relationship between here and here. There is some reasonable relationship where you might actually fancy your chances of estimating leaf area index to a moderate level of accuracy, given some NDVI. But for me, the really key thing about this slide is basically, if you want to estimate a leaf area index bigger than 1 from NDVI, you've kind of got no hope, because any small error in your NDVI here, and it's this kind of region that will give you LAIs greater than 1, any small error, we can bump you up and down this graph hugely. So it's not a good way of estimating leaf area index. Yes, absolutely. It saturates very, very quickly with leaf area. EVI doesn't saturate quite as quickly, but some people really put a lot of emphasis on that, and I would be tempted. Well, it saturates. All the vegetation indices will saturate, some less quickly than others. But honestly, I wouldn't expect EVI to give you a much better relationship than that if you were to repeat the same analysis. It actually bizarrely brings us back to Beer's law and canopy interception, because a good fit to that relationship on the previous graph would be something like that, which is the same form, same kind of exponential form as Beer's law that we saw earlier. So it's probably not unreasonable to say that it is more linearly related to FA-PAR. And if you were thinking about a large spatial area with some mixture of soil and vegetation on it, it's probably related to percent cover as well. And percent cover and FA-PAR in that situation are probably quite strongly related. That's kind of it for my honourable mention of vegetation indices, and so I wanted to cheapen myself a little bit, but I actually think it's important to stand up for them a little bit. So, vegetation indices can provide a relatively clean source of Earth observation data. There's a clear sensitivity in them to the amount and health of surface vegetation, and a classic use is to use them to map phenology, and actually they are probably the best thing out there for doing that in terms of satellite products. But it's not directly related to any particular variables of interest, perhaps most closely to the fraction of absorbed radiation and percent cover. One small caveat that I've put at the end, and then I'm not going to explain it any further, is that actually sometimes they can be useful if you can reasonably expect that you can calibrate a local relationship with them. So if you were to calibrate, for example, a local relationship between a vegetation index and FA-PAR on the ground, then actually it would not be a bad thing to do to use satellite data to scale up FA-PAR over some area based on that relationship. Done with extreme care, making sure you didn't extrapolate between, beyond the land covers for which you'd calibrated that relationship. Okay. Now, this is kind of the conclusions of my talk. How are we doing for time? Because I have two options.
It's like 11:34.
Great. So, passive optical data. It's a very rich source of data, freely available, covering the whole globe on a daily basis. Lots of different spatial and temporal resolutions available. And in that sense, it provides us with a unique tool for trying to understand how the Earth is responding to climate changes, for confronting large-scale vegetation models with data. There's probably nothing else quite like it. Uses the same wavelengths of light as plants do. So, we know that there's some relationship there between what the satellite is seeing and photosynthesis. But obviously, as we've seen, you need to take massive amounts of care to untangle that. And there's all these products that are available that you can use off the shelf in your work. And people do, and people use them well and productively. There's extensive quality assurance information available, which we're going to talk about this afternoon. But the most important thing is to always be aware of those limitations, okay, with those data. Do not use, if you choose to use any of these high derived products, you mustn't use them kind of blindly and naively. There have been some terrible papers published in the past few years, okay, using these data, as well as some brilliant papers. Right. In a way, that's the end of my talk. What I'm going to do at the very, very last bit, actually, let me do that first, and I'll come back and tell you about my... Okay. I've just ad hoc decided to switch the order around. Just want to give you one slide on each of these things, because I don't want you to leave here thinking the only thing in the world is passive optical data. Talked about that mainly because it is particularly useful for sort of the science that all of we do, and because it's easy to get your hands on it. There's also a whole bunch of other stuff that might be useful for carbon cycle stuff. Atmospheric sensors, active optical data. Some of you have probably used LiDAR, actually, which is a great source of data. It's an example of an optical instrument that generates its own source. Thermal data, passive microwave, and active microwave stuff. So many of you will be aware that we now have in space things which can monitor the column average CO2 in the atmosphere. Okay, that's great, and they are starting to become mature. This slide is a little bit out of date. The coverage we have now is really good at these things, and you know, is providing a really relatively unique source of data on atmospheric CO2 concentrations. Always bear in mind, though, that this thing is derived from a measurement of the amount of energy leaving the top of the atmosphere. It is not a direct measurement of CO2. These things never are. So there's a whole bunch of caveats underlying these, just like the ones I've talked about. Nonetheless, exciting. Oh, actually, I should say that's not CO2. That's actually column average methane in that picture. So you can do CO2 and methane and other gases. So remember that it's a column integral. Ooh. That's probably a bit out of date. Does anybody know when OCO is going up? It's probably not in 2013 anymore.
I think it's a bit delayed again.
Yeah, so later this decade. So we have a Japanese mission in space that can do these things. There's a NASA one. The first NASA one failed to launch properly. It ended up in the sea somewhere. It's amazing when you consider how expensive these things are, how many times just the launch fails. But hopefully NASA are putting one up later this decade. That date is speculative at best. LiDAR, which some of you have used possibly, some of you will be aware of. It's most commonly used from aircraft because it's a bit tricky to do from satellites. It's a time-resolved signal, gives you information on things like tree height and canopy structure. Thermal data. I must change this slide because that image is not thermal data. This slide is actually a false color true color composite of some optical data, but it's got active fires mapped onto it from thermal data. So this is an example of a passive technique where the Earth is a black body just like the sun. It has a temperature and consequently emits radiation, but because it's much cooler than the sun, it's emitting radiation at much longer wavelengths. We can deduce all sorts of neat things about the surface of the Earth from that kind of information, and actually a lot of satellites are able to measure in thermal channels, including MODIS. We will have a look at some derived products from those things later today. Passive microwave stuff, sensitive to soil moisture, basically. We currently have SMOS in space, which is the Soil Moisture and Ocean Salinity mission. SMAP, which is the Soil Moisture Active Passive mission, has just been signed off by NASA, so that is going to be launched apparently fairly quickly, but that means, realistically in about 5 years time. But they have to build the thing first. But so there's really that's quite exciting, and lots of climate people are really interested in that particular product. It's a key thing to get right in forcing diurnal cycles of atmospheric models. And active microwave, which is the kind of, if there was one thing I would like to see done better, is that we would have better active microwave missions. The NASA have been basically rubbish at launching microwave missions. The European Space Agency better, but they're fixated on C-band, which is about that long, I guess. Gives us quite nice information about canopies. Really nice because it sees through clouds, not, you know, it's not messed about with much by water vapor in the atmosphere and other stuff. So in that sense, good. But we don't have a really good, long, consistent record. One of the big proposals on the table at the moment is to launch a P-band radar. Now, so C-band, which is what we have most of, is about that length, bounces around inside the leaves, probably tells you something about the leaf area index of the canopy. P-band's like that, I guess. I'm being very deliberately inaccurate here because I can't remember. P-band's like that, and it goes through the leaves, ignores them, and bounces around the trunks. And there's a kind of big hope that it would give us a good handle on biomass, on the standing biomass. Really, it's trunk volume, but you know, that's better than anything else we would have at getting at biomass. There are some huge technical challenges in launching P-band radar missions. So a couple of textbooks. These I'm giving. There's going to be a little folder on all the PCs, and so you don't need to worry about writing that down. The PDFs of this talk and other talks are in that bundle, as well as some papers that I think are worth reading, all by me, of course, and some software, which I'm going to teach you to use later. Right, that's kind of my whole talk. I'm going to give you a really quick, as long as you're up for it, I'm going to talk about some stuff that I've done. Yeah? Right, so a little bit about my research. If I had to give you one sentence that described my research, I would say it's data assimilation of satellite data to constrain carbon fluxes. However, I have this angle on it where I'm arguing very strongly that what we should be doing is, so all the stuff I've taught you about, ignoring it, basically. So I'm making this argument that actually what we want to do is not use those products and use surface reflectance. Now, the problem with that is you have to be an expert in dealing with those things. And so it isn't something that, you know, can just be rolled out, which is why I think it's worthwhile talking about the products and how they're derived. It requires a complex observation operator. And all I mean by that is something that predicts the reflectances, so a canopy reflectance model. But if you read the data assimilation literature, it's called an observation operator. So, can we use MODIS, leaf area index, FA, PAR, GPP, etc., for data assimilation purposes? And you're all going to learn what data assimilation is during this week. Yes, you can, but we've seen all those things that are, you know, bad about them. And key, actually, data assimilation really relies very strongly on having well-characterized errors in your observation and your models. And we don't have that from the MODIS products. It's really difficult to characterize those errors. So it makes them almost impossible to use really well in a well-functioning data assimilation scheme. I also think that we have all these assumptions underlying those products, especially the photosynthesis products, about the kind of modeling and response to temperature, the climate data that drives those things, that will disagree with our models. And if you assimilate those data, you're also assimilating all those assumptions into your model. And they may just not be compatible with what's inside your model. So I think on a very kind of philosophical level, it's just the wrong thing to do. My starting point, so when Dave said I was the first person to work with Earth observation data and ecosystem models in a data assimilation sense, this is the model I chose to use. A guy called Matt Williams had published a big data assimilation paper in Global Change Biology with this model a few years before, so it seemed like a good place to start. The advantage of this model is that it is really simple. It's like 15 equations 15 lines of code, actually. Okay, it's not, so SIPNet that you'll use later in this week is a simple model, but has a few hundred, maybe a few thousand lines of codes in it. I'm not sure. Have a look, you'll see the code. This is really short, but the advantage of that is it's numerically incredibly efficient. And we can run lots of experiments and do lots of very numerically complex things. It's not really, in my opinion, a great model for, you know, you couldn't do anything prognostic with it. And without confronting it with lots of data, you probably won't want to do anything diagnostic with it either. So in its own right, it's not a useful model, but it's specifically designed to be extremely efficient and to be used in data assimilation. In that sense, it can be very useful. Okay, so took that model as a starting point. And the first thing to think about is basically what I've already explained. Should we assimilate those high-level products from satellite data? And I think I don't need to go over this because I've already explained it. But basically, we have assumptions in those things which disagree with the assumptions in our models. So I argued we shouldn't do that. We shouldn't take those products and assimilate them. But the flip side of that is we need to make our model predict our observations. That model and all other ecosystem models, like I was explaining at the beginning of this lecture, are not capable of predicting those basic satellite observations of canopy leaving reflectance for a whole number of reasons. So to do the data assimilation without using those products requires the addition of codes to that model to allow it to predict the satellite reflectances. This is actually quite commonly done in numerical weather prediction to assimilate radiance data from satellites. And the technical lingo they use is to call that thing an observation operator. And in our case, our observation operator is just a canopy reflectance model. What it allows us to do is keep our assumptions consistent between our model and our observation operator because we have complete control over that system. We then use those observations to compare against, we use those predicted observations to compare against our actual observations and allow our data assimilation scheme to adjust our model on the basis of those observations. Don't worry about all that, those awful acronyms there. You'll all be experts by the end of the week. I started using that semi-discrete representation I talked about earlier, which basically the canopy looks like that, which is not far off what most ecosystem models look like. And this experiment, we were working on the Oregon transects. And so the forest in this area was not very homogeneous, basically. And this represents a homogeneous canopy, and we couldn't get it working. So we stepped up the complexity in our observation operator and switched to something which still contained those radiative transfer components, but now clustered all that green goo into discrete crowns. So it's just like the equations I showed earlier, but now I have separate routines for working out how much radiation completely misses it and working out how much of different shaded and illuminated components of the vegetation and the soil that the sensor can see. Okay, so it's quite, in terms of complexity, this model is really a bigger step up from what we've been talking about, but it turned out to be necessary. My proof for this, and I apologize for the very small lettering on these plots, is the match to the observations, but specifically the way they are able to represent the angular distribution of radiation. Both, this is the red channel of MODIS, this is the near infrared channel of MODIS. X-axis is the viewing zenith angle in both cases, and the y-axis is of reflectance. The blue dots are observations from the satellite, and the red dots are predicted from the observation operator that we bolted onto the ecosystem model. Now, actually, the key thing about this is that the one-dimensional plane parallel canopy cannot do both of these shapes at the same time because the canopy is sparse. So we have to go to a geometric optic representation to be able to correctly predict our observations. Don't worry if those last two sentences were completely meaningless to you. It's probably not important, and I'm very happy to explain what I was talking about later. This is the money shot, though, if you like. X-axis in both cases is just the day of year for, I forget now, 2003, something like that. Black dots are eddy covariance estimates of net ecosystem productivity using the one of the Flux towers on the Oregon transect. The black dots are identical on in both panels. They're in exactly the same position. And importantly, they have not been anywhere near the model. They are just there for comparison. So it's like a validation of what we've done. In this top plot is the model trundling along on its own, being forced by local meteorological data, but with no observations being assimilated into it at all. We're using a technique called the ensemble cam filter, which you'll learn about from Andy Fox. And the red line is the mean of those ensembles, and the spread around it represents one standard deviation of those models. Nice technique because it gives us a really good handle on uncertainties coming out of things. The bottom panel then, I'm assimilating reflectance data. So only reflectance data into this model. Nothing to do with carbon as such. And the nice thing is that we get the day-to-day variability in our NEP much better than the model without any data assimilation into it, which, you know, in many ways was quite a surprising result. But there's a really important thing I want to bring out about this. There are things like this group of dots here that we just don't capture. And probably, and somebody in this room can tell me better, so can explain this better than me, this is probably due to flushing of new leaves and the use of labile starchy carbohydrates, I would guess. Okay, that would be my hunch. Let's just say for the sake of argument that is what's going on. And we don't capture those inside the model. So what we can't do, if your model doesn't have those processes in, then there's nothing we can do. It's not magic data assimilation and using satellite data. Those processes are missed because these data are missed because the processes are absent from the model. But that's a nice thing about using data assimilation, is it helps you narrow down on those processes you might need to represent different observations. Another good thing about having done this is that because the satellite data is this big, allows us to cover wide areas, we can spatialize those fluxes in a way that you couldn't do using the model alone. So we did it's quite computationally intensive, even though we were using a simple model. So we did a small area, and that's the Flux tower. That's where the data on the previous slide comes from. But we estimated it over a 4.5 by 4.5 kilometer area. And the nice thing about this is that the NEP, the carbon balance, roughly corresponds to what you might expect things to be doing given the land cover that's there. So this top corner where the NEP is getting closer to being in balance is a mature part of forest on the Oregon transect. So we have actually quite a lot of faith that this technique was working well. That is it.